Hello guys, this is Malinki. Welcome back to my channel Voice of Malinki. Today we will discuss about the complement system part 1, that is classical pathway. So if you are new in my channel, please subscribe my channel, and if you like my video, please do like, comment and share my video. So let's discuss classical pathway. Now the complement system consists of 3 pathways. Among them, first one is the classical pathway. So today we will discuss about classical pathway. The complement system is a part of immune system that clears microbes and protects our healthy cells. So complement system is a part of our immune system of the body that clears microbes, that kills pathogens, and therefore they protect our body, they protect our healthy cells. Classical pathway begins with the formation of antigen-antibody complex. Okay, so when a pathogen enters our body, it is supposed to be the antigen. So the microbe is the antigen. An antibody binds that microbe. When this antibody binds that microbe or binds that antigen, the antigen-antibody complex forms. And when that antigen-antibody complex is formed, classical pathway of complement system begins. So, complement system consists of number of complement proteins. Huge number of complement proteins are found in our blood, but they are staying in the blood in an inactivated state when our body is healthy, when our body does not have any infection. Now these inactivated complement proteins are found in our blood that could be activated as a result of antigen-antibody complex formation. So when antigen-antibody complex is formed, the complement proteins that were inactivated in the blood would be activated and they will start their action. The formation of an antigen-antibody complex induces conformational changes in the Fc portion of the antibody molecule. So we know that an antibody contains 2 parts. This part is Fab region that binds the antigen. And this part is the Fc region. So this part is open. Now, in classical pathway, 2 types of antibodies actual function. They are IgM and IgG. So IgM and IgG antibodies undergo the classical pathway. And when an antigen-antibody complex is formed, it induces the conformational changes in this Fc portion that exposes a binding site for the C1 component of the complement system. So complement system contains huge number of proteins and these proteins are named as C for complement, and then we put numbers like 1, 2, 3, 4 after the letter C. So among them, C1 is a complement protein that will bind the Fc receptor of the antibody that is bound with antigen. Now this C1 protein is a macromolecular complex. Why it is a macromolecular complex? Because it contains more than one molecule, consisting of one molecule of C1q, two molecules of C1r, and two molecules of C1s. So this C1 is a macromolecular complex and it contains different molecules. Among them, one is the Q, two are R, and two are S. These are different protein subunits and they are held together in a complex. Hence, it is named as C1qR2S2. Here, Q is single, pair of R and pair of S subunits are present in this complex. Now, binding occurs between C1q and Fc portion of the antibody. So, if this is the antigen and this is the antibody and it is the Fc part of antibody, we know that the binding between the Fc portion of antibody and C1 molecule is now done. But C1 molecule is a multi-protein complex and the Q subunit of C1 actually binds the Fc portion of the antibody. And this binding activates C1R. So C1 has pair of R subunits and pair of S subunits. So this binding, that is binding between C1q and the antibody, this binding activates C1R, and when C1R is activated, it will activate C1s. And this C1s, it is a protease enzyme. What is protease enzyme? That means it cleaves proteins. So C1s can cleave other proteins. It activates and cleaves C2 and C4 complex. So C1s can cleave C2 and C4 protein because it is a protease enzyme. Now C4 is cleaved into two fragments. One is C4a and other is C4b. This C4b, it binds to the microbial surface, whereas C4a is released. And C2 protein is also cleaved by C1 and it is cleaved into again two fragments. One is C2a and another is C2b. So C2a binds to the C4b and C2b is released. We can understand it better in the diagrammatic representation. So here we can see that antibody binds the C1 complement protein, and this antibody is binding the antigen here. Then this C1 will cleave C2 and C4. C2 is cleaved into two fragments C2b and C2a, and C4 is cleaved into two fragments C4b and C4a. C2b is released, C4a is released, C2a and C4b, they will combine to form C4b C2a complex. So it is again a protease. This C4b C2a again is a protease, and that can cleave other proteins. And it is known as C3 convertase. Why it is known as C3 convertase? Because it can convert C3. It can activate C3. C3 is one more complement protein. So it activates C3 protein, and C3 is the most abundant protein of the complement system. Please try to remember it that C3 is the most abundant protein of the complement system. Now this C4b C2a complex, that is called C3 convertase, it will cleave C3 into two fragments. One is C3a, another is C3b. So C3a is an anaphylatoxin. So what is anaphylatoxin? So it goes and binds to the surface of basophil or mast cell. So when C3a is formed, it will go and bind to the basophil or mast cell surface. So it is basophil or mast cell. Now C3a will bind this basophil or mast cell surface. And when the binding occurs, they will release histamine. So histamine are released from the basophil or mast cell after the binding of the cell and C3a. This histamine induces vasodilation and increases capillary permeability. So what is vasodilation? Vasodilation is nothing but the increase in the diameter of blood vessel. So the blood vessel diameter is increased, that is called as vasodilation. And also histamine increases capillary permeability. That means the blood capillary is more permeable. That's why it permits some immune cells to enter at the site of infection from the bloodstream. You can understand vasodilation and capillary permeability from my lecture, that is inflammation. I have explained these two phenomena there very well. So please have a look on that. So as histamine induces vasodilation and increases capillary permeability, it promotes inflammation because vasodilation and increase in capillary permeability ultimately promote inflammation. Therefore, immune cells are capable of reaching at the site of infection because we know that inflammation is required for the immune cells to reach at the site of infection. Now C3b. So when C3 is cleaved, it produces two fragments C3a and C3b. So now we know that C3a is an anaphylatoxin and it works as anaphylatoxin and it induces inflammation. But what does C3b do? So C3b acts as an opsonin. It enhances phagocytic process. C3b enhances phagocytic process because it is an opsonin. I have prepared a separate video regarding opsonin. So if you don't know about opsonin, please go and check that video. And C3b also binds another complement protein, that is C5. So C3b is not only an opsonin, it binds another complement protein, that is C5, and creates a conformational change. So this is C5 and C3b binds this C5 and creates a conformational change on that C5. This conformational change is required because it prepares C5 to be cleaved by the C3 convertase or C4b-C2A complex. So we know that C3 convertase, that is C4b-C2A complex, it cleaves C3, but it also cleaves C5, and the C5 is required to be changed to undergo the cleavage, and that is done by C3b because C3b creates a conformational change on C5 that helps in the cleavage process. So C3b and C4b-C2A, that is the C3 convertase, these two are known as C5 convertase because it is now converting C5 or it is now activating C5. So C5 is now cleaved by C5 convertase into C5A and C5b. So C5 is cleaved into C5A and C5b. So C5A is again an anaphylatoxin. So we know what an anaphylatoxin does. I am not repeating the thing. So it also acts, C5A also acts in chemotaxis. That means it attracts other immune cells at the site of infection. Because we know when an infection occurs, it requires huge number of immune cells to come at that spot. And that is done by the chemotaxis. So C5A helps in that chemotaxis. And C5b, that is also released by the cleavage process, it contributes in the formation of membrane attack complex or MAC. So membrane attack complex or MAC is very important and it is the ultimate thing in complement system. So you need to understand the MAC or membrane attack complex very well. So this C5b contributes in the formation of membrane attack complex. Now how does this MAC form? C5b, that is released from C5, now binds to C6, C7 and C8 molecules and creates C5b, 6, 7, 8 complex. So C5b, C6 and C7 and C8, these molecules will create a complex that is C5b, 6, 7, 8. And this complex is attached with the surface of microbe. So this complex is attached with the microbe surface. And this complex induces the binding of C9 molecules. Number of C9 molecules will be attached to the cell surface. Which cell surface? Surface of microbe. So, in the plasma membrane or in the cell surface of microbe is now occupied by C5b, C6, C7, C8, C9 molecules. And these molecules are forming the complex that is known as MAC or membrane attack complex. But why it is named so? So this MAC creates a pore in the pathogen surface that fills the fluids into the cell that leads to osmotic lysis of the microbe. We can understand it better in the diagrammatic representation. So here you can see, this is the bacterial plasma membrane. This is the bilayer, bacterial plasma membrane bilayer. Here C5b is attached first, then it recruits C6, C7, C8, and then they will recruit number of C9 molecules. We can see here number of C9 molecules are there. They will make the MAC, and these C9 molecules will make the pore, or you can say this is the channel or pore in the bacterial plasma membrane. It could be any other microbe also. When this channel is formed, water or fluid can enter to the cell cytoplasm from outside. This is cytoplasm. And when fluid enters into the cytoplasm of a cell, the cell will burst because of the osmotic pressure. So the cell lysis will occur. And in this way, classical pathway kills the pathogens or microbes that have entered our body. So this is all about the classical pathway.